Kia ora, everyone. My name's Colin. I am a PhD student at Victoria University of Wellington, and I'm supervised by a quite diverse range of people from genomics, Aotearoa, and plant and food research. But I'm not going to go too much into that. You can look me up for that if you'd like. I'm going to speak to you today about conservation genetics of a Myrtaceae species in New Zealand, maire tawake, also known as Syzygium maire, and hopefully just give you an idea of what research I'm doing. It's going to be quite a sort of quick blast through it all, and then any specific questions or anything, please feel free to contact me. I can't see who's in the audience, but I'm sure that many of you have heard me speak before or had chats with me, and I look forward to continuing those. So just to start, Syzygium maire or maire tawake or swamp maire or waiwaka, these are all sort of the more traditional names for it, as opposed to the more Western Syzygium maire name, and it differs depending on where you come from. The species is a major component of the North Island swamp forest or Te Kao Maui. It occurs across the North Island, mostly restricted to the western side, as well as the northern tip of the North Island, South Island down here. It's an important food source for native birds, and it's the only endemic Syzygium, despite there being about 1,000 Syzygium species worldwide. It's the only one in New Zealand. It's taonga, which means that it's sacred or treasured by Maori, and it has many uses traditionally for them, including treatment of various diseases such as ringworm as a food source, and it's highly durable wood. Ecologically, this species is an important umbrella species within wetland ecosystems, as you can see here. At a certain level of waterlogging of ground, maire tawake becomes a dominant species in an ecosystem, and considering that much of New Zealand is waterlogged, much of lowland New Zealand is waterlogged, the habitats in which this species would have been having an important umbrella role would have been vast. The species is, however, nationally critically threatened. So this is one of the highest threat levels, if not the highest threat level in New Zealand. And this is due to two major reasons. Firstly, historically, the species has undergone large-scale range reduction. So specifically, as I said, it occurs mostly in lowland habitat. Much of this has been drained in New Zealand. Across the world, this sort of habitat has been drained, but especially in New Zealand for pasture use, resulting in it losing over 98% of its habitat or wetland forest in certain regions in New Zealand. So I think across Aotearoa, over 70% of forest has been lost. Most of this has been restricted to lowland areas, and much of these lowland areas were wetland. So for instance, in Taranaki, it's sort of, Bill Clarkson said that there was something like 79 hectares of land, there would have been 76, which were almost dominant maire tawake forest. However, this is all now swamp, is all now pasture land. Also of importance is that there's no seed bank. So these seeds are both recalcitrant to preservation, as Karen van der Walt and Roger Ben and others have found. But also the seed that is produced in a season needs to germinate that season. There's no seed bank in the ground. So once trees are removed from an area, the only way that they can recolonize is through seed dispersal from neighboring populations. And this then brings us to the last point, is that these populations are now highly fragmented. So many of them are just small remnants, such as this patch in Taranaki, which are surrounded by non-natural matrix, such as pasture land or urban areas through which seed dispersers, such as birds, wouldn't be able to move or wouldn't as readily move. So you're getting possible genetic isolation of these small patches, and this could result in inbreeding and so on. The other major threat is Myrtle Rust. That's why I'm speaking at this webinar. This species is extremely susceptible to the pathogen. Infections have been observed nationwide. I've seen them everywhere from the far north to most recent reports, so in the Wairarapa and so on. And all aerial tissues, flowers, fruit, and seedlings are affected and severely affected, which possibly could lead to reproductive failure. Although this hasn't been proven, it's highly likely. I've seen entire stands of thousands of trees where almost all seem to have large-scale infection, and then functional extinction of the species. So really, we need to restore the species. There's a huge amount of interest in restoring the species, as is there a huge amount of interest in restoring natural environments across Aotearoa. But we needed to be able to do it in a planned manner. So firstly, we need to be able to conserve germplasm in a way that captures the entire genetic diversity of the species. This is because really we can't go and do many large-scale infection or so on trials for many native species because it's just too expensive. So we sort of want to preserve as much genetic diversity as a proxy for potential adaptability in future to disease, to climate change, and so on. And then possibly we can then use those stocks to do infection trials in common gardens and those sort of experiments. And then secondly, we need to choose sites that can mitigate environmental risks and fragmentation. So my work primarily deals with the first question. The second question is being explored by my supervisor, Julie DeLip, and some of her collaborators, Stephanie Tomsher, and so on. And they have a paper that's in submission, which has gone and mapped out some potential restorable refugia in the Wellington region and has found that even when considering myrtle rust, there's potential for protection of the species, especially when coupling that with restoration projects. So making sure that sites are accessible and able to be treated with fungicides if needs be. But hopefully we can get them in to speak about that at some point. So really, the first introduction to my major question is the great eco-sourcing debate. So in New Zealand especially, there's the principle of eco-sourcing, which is being reverberated through many restoration projects, which is that we want to source seed as close to the site of restoration as possible. And this is assuming that local seed will be adapted to the local environment. However, there's no real genetic evidence for how big these boundaries should be. How close is too close? By doing this, are we selecting from a limited gene pool, which has already seen huge amounts of genetic loss and therefore exacerbating inbreeding and founder effects and so on? And then also, how far is too far? At which point are we diluting local adaptive genetic variation? And of course, one size doesn't fit all species. So a recent paper by Peter Heenan, which deals with eco-sourcing in New Zealand, has proposed really large-scale eco-sourcing boundaries. So these are on a regional scale where we have, this is mostly looking at Kanuka research, and this is a wind-dispersed species, so it's large, so it's going to have less variation over larger areas, versus maire tawake, for instance, which is bird-dispersed and is likely to have more genetic variation or more genetic structure across smaller areas. So Peter Heenan found that there are these large eco-sourcing boundaries across the North Island, just 3. Whereas DOC and so on and other organizations had often purported something like within a kilometer of your restoration site should be the maximum sort of distance you go for restoration. However, according to Peter Heenan and others, and what I found in my research, this is probably too close. So just a quick aside, I'm going to blast through this so that I have time for the other stuff. The first thing we did was construct a reference genome for the species. So this is one of the first sort of reference genomes of New Zealand tree species. There are a few others, but not too many. We looked at a single individual outside Palmerston North, along with Rangitangi o Manawatu, which is the local iwi. They're acting as kaitiaki or guardian of this genome and are undergoing a naming process currently. This is a whole nother side, but a lot of this project has been working closely with iwi and making sure that things like data sovereignty and such are respected, which I think has been really fulfilling area of this project. We did PacBio hi-fi sequencing, which is a long read, high accuracy sequencing sequencing platform, and got some really good results. So we got scaffolds, which is pseudochromosomes, it's lengths of sequence, which represented entire chromosomes. So that's 41 megabases, which in previous technologies would have been unthinkable. And the biggest three represent entire chromosomes from telomere to telomere. We also have high gene representation for these and other completeness scores, which show that we really have constructed a high quality genome for a low price. I think it was $5,000 US or somewhere around there. So not the smallest amount of money, but in terms of what could be done a few years ago, really, really promising. I'm going to skip over that slide. So back to the question of how diverse is maire tawake across Aotearoa. To answer this question, I went across the country and sampled 301 adult trees. So this was from Northland, Bay of Plenty, Taranaki, Palmerston North, and the trees surrounding the genome, the one we sequenced for the genome, Wellington, and then the furthest for the population in the Marlborough Sounds. And this, once we sequenced these via low coverage whole genome sequencing, something like 10x coverage, we got 1,000 SNPs. So this is 1,000 points, I mean, 1 million SNPs. This is 1 million points in the genome where each of these trees may differ to each other. So this is a really large data set in which we can compare how different trees from one site to the other are. So the first finding here was that national population genetics reinforces the large-scale eco-sourcing boundaries that have been suggested by Peter Heenan and others. So Waipoua represents Northland, Tauranga, Bay of Plenty, Taranaki, Manawatu, and Wellington region, and Marlborough. Each of these panels, K is a clustering algorithm, so that suggests how many clusters or how many common ancestors each of all of these populations may share. So in the most simplest explanation, we see that if they shared 3 common ancestors only, the Manawatu and Wellington populations all look like they mostly have this blue ancestor or light blue ancestor. Waipoua or Northland is derived mostly from this orange ancestor. Taranaki and Tauranga are a mixture of the two, and Marlborough region is once again largely dark blue. So even as you increase the number of potential ancestors, you see that this clustering remains similar, where Manawatu and Wellington look like they are the same sort of, have the same sort of ancestry. Northland and Marlborough look quite distinct. So just to hark back to that map of New Zealand that Peter Heenan has published, once again, Wellington and the Manawatu look like they're their own eco-sourcing region. Central North Island and Northland, each of their own, and then the tip of the South Island. FST, which is a measure of population differentiation, was really low in Peter Heenan's study. And once again, I think that's possibly because they were looking largely at Kanuka, which is a wind dispersed species. So populations are not very different to each other. Whereas in our study, we found an FST of 0.35, which shows that populations are quite a bit more different to each other. Now, what this suggests is that maybe, although we are seeing these large boundaries, we might want to sort of delve into each one of these populations and really narrow that down. Potentially still sourcing a tree from Palmerston North and planting it down in Wellington wouldn't be the best idea. I'm going to skip over this. No, I won't actually. This is kinship for all of those trees that I just showed you, so how related they are to each other. And this once again just reinforces that idea where Northland trees are least related to all the other populations. And as we move further south, we see higher degrees of relatedness. This might suggest that the Northland trees are the most genetically diverse, potentially where there was a remnant population in glaciation or the original founding populations, and then trees moved south as conditions became more favorable. The Marlborough population is extremely highly related and inbred, which might suggest that that was from a recent founder effect. It's also a really small population in a bay surrounded by quite high hills, so it's possible that it's been isolated for as long as it's been there. Looking at local scale analyses, we can flag individuals that harbor unique genetic diversity. So in this panel, we can see pairwise FST. So this is how different each individual is to each other individual in each of these sites. So these are sites within Palmerston North area that I sampled. And we can see that sites within sites, trees are largely similar. So that's a value of 0, where a value of 1, which implies high difference, occurs more often between sites. So this might suggest that eco-sourcing boundaries might be smaller within areas, and that we might not necessarily want to always dilute the genetic variation within sites from trees outside of, with plantings from outside of those sites. However, we need to look at this in a larger scale, in a larger context, and compare this to other populations as well, because these might look very different when comparing directly to each other, but when comparing to populations 10 kilometers or 20 kilometers away, they might not look all that different. We also see quite a high heterozygosity, 0.29 within this population, which suggests that there is a fair amount of genetic variation. And just one last application of this is we have sequenced we've sequenced 55 seedlings from one of those sites previously. Once again, all thanks to Rangitane o Manawatu. These seeds, there were two reasons for doing this. One was to test whether genetic information could inform seed sourcing projects in future. And another was to start looking at whether there are any sort of genetic associations with phenolic content. So I'm not going to show those results today because we've still got to look at that. But for each one of these seedlings, we've characterized the phenolic content. So these are compounds that are often associated with resistance to various pathogens, including fungal pathogens such as Myrtle Rust. And we've got genetic variation for each of these, as well as variation in phenolics. And we're hoping to go and try and understand what the genetic basis for variation in those phenolics are. And then potentially we've got a common garden, maybe do some infection trials and try and figure out whether there's any association between those three variables. Just in terms of this seedling set, it's allowed us to do two things just right off the bat for planting strategies. Firstly, often seed sourcing takes place in a way that nursery managers and that go to a single tree which is producing a lot of seed and collect extensively from that, go and propagate that in their nurseries, then plant out entire regions with seed from a single mother. This is once again a kinship analysis, so it's showing how related trees are, as well as how inbred they are. If you look on the axis where self versus self is how inbred a tree is, and we see that this block is highly related and a lot more inbred than these trees down here. And this is likely a block that was sourced from a single mother tree. And if we went and then planted out an entire restoration project, we would get highly inbred trees in that site, which would then go on to result in possibly lower adaptability to climate change, to infection risk, and so on. But with genetic information, we can understand that there are less inbred trees within this population and seedlings, and ones that might hold a higher amount of genetic variation, which we can then go and propagate as well, so that we can make sure that there's a fair distribution of different genotypes within the seeds that are getting planted out. Secondly, this allowed us to identify contaminant seeds that weren't sourced from the local rohe or region, and would thus have potentially contaminated those genetics of the local trees. So just in conclusion, I've managed to construct a complete and accurate reference genome, and these can be quick and cost effective for endangered and rare species. I've identified 4 to 5 distinct populations of maire tawake, which are approximately reflective of those eco-sourcing boundaries proposed by Heenan et al. FST suggests that boundaries may need to be more local in this species, and I'll look more into that and see if we can tease apart at which point we might want to draw those lines. And it also suggests that while restoration projects could consider broader scale eco-sourcing zones, you might want to inform them at a more local scale by local context or values that are important to the people that occupy that land, such as Mataranga and so on. So what this could look like is potentially bringing in a small amount of seeds from further afield, which would then allow the genetic diversity of those local populations to be bolstered, but still keep most of it within that local area. And thank you for taking the time to be here. I did, there it is. I have my email up there if you would like to contact me. And for now, I'll take some questions. Thank you.
Thanks, Colin. That was awesome. So there's already some questions coming in. Audience, if you have any other questions, please put them into the chat panel, and I will bring them to Colin's attention. So the first question is that we're starting a wetland restoration project and only have one maire left in the 250 hectare area. Would the seeds be fertile, or do you need male and female trees?
As far as I know, the seeds, the plant is able to self-pollinate. So there's a plant in Nelson, which Karen van der Walt had collected seed from before, which is also isolated. I think the nearest ones are over in Marlborough Sound somewhere, and that seed has all been viable. So I imagine your seed would be viable. Just be aware that if it's self-pollinated, those seedlings are almost all going to be sort of not quite clonal, but they're going to be very highly related, inbred, and you might want to go a bit further afield just to get some seed if you can to plant there as well, just to make sure that there is some genetic diversity in your restoration project. But I'm happy to speak to you, and we can look at maybe some of the samples that I've taken and see where your restoration site is and see if we can work out where best to potentially source some extra seed from, if you'd like.
Sounds good. And that answers it. The second question, which was whether they should bring trees in from the outside of the catchment area. So that was nicely done. So the other question that I had was that looking at that FSD value that you had, do you see a similar sort of FSD value in other animal dispersed seeds, or trees that have animal dispersed seeds, or is this pretty high because it's such a restricted population?
Yeah, so I'll have to go and brush my knowledge up on New Zealand trees specifically, but I know that there are some higher FSTs for genetic differentiation for some New Zealand trees, such as Aotearoa and that. But in general, in like the more global literature, trees that are more animal dispersed would generally have higher genetic variation. The highest sort of ones are observed in rainforest trees that are pollinated by small insects and dispersed by small animals, because the seeds just don't move very far from the adult trees. So you get these sort of really local scale subpopulations where everything's really highly related, versus wind pollinated trees where pollinated or seed dispersed trees where these can be dispersed widely across the landscape.
It's interesting. And with the I guess with the genome that you've done for the species, are there other genomes of Syzygium available, and how distinct is it from other Syzygium species?
Yeah, it's sort of one of the things. It's like I've tried to fit in a lot here. I could have spoken at length about any one of these things. Yeah, so we'll be publishing. I'll publish the genome before the end of this year. That's on the cards. And in that paper, I have compared it to Syzygium aromaticum, which is clove, and Syzygium grande. Um, Both of those have genomes published, and all in all, they all look very similar. It doesn't look like there's a huge amount of genomic
Mm.
variation between the two. In terms of small scale variation, I can't tell you, but in terms of large scale, they all have 11 chromosomes. There aren't huge amounts of duplication. They're all roughly the same size. So even though there are 1,000 species of Syzygium, it doesn't look like there's a huge amount of large scale genomic variation between them.
And where is the center of diversity for Syzygium? Is it Australia or is it somewhere else?
Yeah, it's around. I think Australia has the most species. I stand to be corrected. It's around sort of the Pacific area is where there's most Syzygium, but they occur in Africa and elsewhere as well.
Sounds good. Well,
Primarily sort of rainforest trees. Yeah.
yeah. No, it's really interesting. It's also really interesting that that New Zealand only has one species. Of course,
Mm.
we have, as you mentioned, we have a lot of introductions from other places, but yeah, it is very interesting. Um, so I guess if that's all the questions we have, thanks again, Colin, for your presentation. And a video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days. It's also going to be emailed to everyone who registered. Our August webinar is just around the corner. We'll hear from Karen Somerville and Veronica Viola from the Botanic Gardens of Sydney. It's on Wednesday, August 9th, and they'll be presenting on ex situ conservation to support recovery in the wild. Keep a lookout for the webinar invite in the next couple of weeks. Thanks, everyone. Haere ra. See you